Our body constantly has to defend itself against harmful bacteria, viruses, or internal threats such as tumors. Luckily for us, we have a powerful protective mechanism called the immune system. To better understand how different diseases affect us, it's thus of crucial importance to study this immune system. I typically study the mechanisms of the immune system, but by analyzing biological data, which I showed here as this blood sample from a patient. And then this data I typically analyze with computational tools to extract knowledge from it. Since today I will be talking a lot about the immune system and some computer concepts, I will first present a short introduction in which I'll briefly go over how the immune system works and what the different parts of a cell are that we are actually interested in. And then I also introduce some computational concepts of how we can teach the computer to learn from the data or with extra information. After this introduction, I will go over 3 projects that I worked on. The first one is Tenga, a tool that I developed to identify trajectories, and then 2 biological applications on memory cells and mechanisms of tolerance. But first, I will start with this introduction and a small peek into how the immune system works. Viruses can enter our body and they can infect our cells. When they do, the infected cells become kind of factories that can start producing new viruses that can then infect other cells in our body or even other people. This is when the immune system steps in. First, these immune cells that are called dendritic cells because of their shape will recognize the infected cells and they will send an alarm signal to the whole body. This results in a strong inflammatory state in the whole body, which can explain why we sometimes experience fever or fatigue after we've been infected by a virus. Several cells from the immune system will answer to this call, and today we'll focus mainly on T cells. After they have been activated by these dendritic cells, they will typically start to multiply and they will start producing destructive molecules such as the perforin. Once they are ready, these cells will then migrate to the site of infection and they will recognize the infected cells. When they do, they will send their destructive molecules at them, which will have as a result the destruction of the infected cells. After the infection has been removed, memory cells will patrol in our body and confer us a long-lasting protection against this same virus. So in summary, in this first part, we've seen different actors of the immune system. We've seen these dendritic cells, whose role it is to detect the pathogen and send an alarm to other cells. And we've also seen these T cells that when they get activated, they can either multiply and kill the infected cells, or they become memory cells that can patrol and protect us. I'll now move to this second part of the introduction and mention briefly which parts of the cell we focus on. So just as a quick reminder, this is a eukaryotic cell. We have the nucleus here in which we can find the DNA. And if a cell wants to start expressing some genes, it first has to make a gene copy that can move out of the nucleus and can then be used to make proteins. These gene copies are actually what we're interested in. I typically work with RNA sequencing data. This is useful if we want to compare cells, for instance. As an example, we want to compare these 2 cells and we can see that we find different copies of genes in them. In the first cell, we find 3 copies of this perforin gene, but 0 copies of this red gene. Whereas in the second cell, we can find 0 copies of the perforin gene, but 2 copies of this red gene. And so just by looking at what genes these cells are expressing, we know, we have a hint that this first cell is probably preparing to fight, and that this second cell is doing something else. I would now like to introduce some computational concepts and first show how we can teach a computer to learn from the data. For this, I'll take as example this object, this red star, and we can describe this object using several variables. In this case, we use 3 variables: the shape, the color, and the size. So we know that this object has the shape of a star, it has a red color, and it's relatively big. We can describe these other objects in exactly the same way. The shape is either a star or a circle. The objects are either red or blue, and they are big or small. Then we can teach the computer to group similar objects together. And in this case, it would make 2 groups. First group with these red stars that are very similar, and the second group with these blue objects. But what happens if we show the computer this object? Well, it is still, well, pink, which is quite close from red, and it still has kind of the shape of a star, so it would probably end up in the group 1. And if we look at the second object, we can see that it is almost blue and quite roundish, so it would probably still be assigned to this group 2. But at some point, we might encounter an object that would be the exact mix between the original red star and blue circle. And in this case, you see that it doesn't really make sense to try to talk about groups anymore, but we're actually rather seeing a transition between objects. At this point, we will talk about a trajectory, which is a very powerful tool to describe gradual transitions between objects. A trajectory is also a nice tool because it allows to model these changes and to see which features, which variables really change along the trajectory. For instance, the color really changes. It gradually changes from red to blue, and the size gradually decreases along the trajectory. So in summary, we've seen that we can teach a computer to learn from the data, and it can either learn how to group objects into groups or how to identify trajectories in this type of data. I will now move to the last section of this introduction, which will show how we can teach a computer to learn with extra information. Here, I have represented the extra information with these 2 boxes that contain different objects. And the idea is to teach the computer to learn the differences between these objects. Because if we do this correctly, then we can present new objects to our classifier, and it will be able to assign the objects to the correct boxes on its own. This might look like a quite trivial task. However, it gives us a very powerful tool. For instance, if I have these images of patients who have either pneumonia or are healthy, well, if I showed you this image, you would probably have issues telling me to which group it should be assigned. But we can teach classifiers to do this in a very efficient way. Actually, nowadays, we have classifiers that are able to classify these images better than real doctors. So in this introduction, we've seen different aspects of how the immune system works and what parts of the cell we actually focus on. We've also seen how a computer could learn directly from the data or with external information. Now I will move to these projects that I mentioned at the beginning, and first on Tenga, a tool to identify trajectories. Because in my introduction, I've shown you that we can extract trajectories from this type of data, but I didn't tell you how this happens. And this is a process that happens in 2 steps. First, we need to find a nice representation of the data, and then we can look for a trajectory in this representation. I will stop working with red stars and blue circles and actually show you the data that I actually work with, which is these cells in which we measure different copies of genes. If I wanted to represent this data, I could, for instance, create a new space in which the x-axis reflects the amount of purple copies, and the y-axis reflects the amount of red copies of a gene that are inside a cell. In this case, these 2 first cells would be represented as this in this space. Now, if I want to represent this 3rd cell, I need another dimension to also represent this green gene copy. And I will quickly run into issues because I need one more dimension every time I want to show a new gene. And since humans are not too good with visualizing things in more than 3 dimensions, we need to come up with a different visualization. This we do by computing distances between cells. If I here take a Manhattan distance between cells, I see that these 2 cells are different because the first one has one purple copy that the second cell doesn't have. And so the difference between the 2 is 1. Then I can also look at the difference between the second cell and the 3rd cell. Again, the only difference is this green gene. So again, the distance between the 2 cells is 1, and so on. Then I can also compare cells that are more different. For instance, the 1st and the 3rd cell, they have one purple and one green copy, gene copy of difference, so they have a distance of 2 between them. And in this way, I have defined the distances between all my cells, and I simply can represent these distances in a new space. So here I respected all the distances of 1 or of 2 between cells, and you can see that I'm visualizing in 2 dimensions the information that was carried by all of these genes. I showed only 4 genes here, but this works for as many genes as you want. So this type of visualization allows us to visualize the information carried by thousands of genes in a very reduced number of dimensions. So once we've learned how to represent the data, we can then look for a trajectory in it. Of course, I typically don't work with 4 cells, but thousands of them. And in this representation, each little black dot corresponds to a cell. Typically, we also work with data that is much more messy, but for the sake of this presentation, to keep it simple, I will take this data set that is very clean. And so now we want to find a trajectory, and this is where I would like to introduce Tenga, a tool that I developed to identify trajectories. Here is briefly how it works. It will be initialized by randomly placing 2 nodes in the space that are linked by a blue line that I represented here. Of course, this blue line doesn't represent the data very well, so it needs to be adapted. And this will happen by selecting one point in the data, for instance, this one, and this point will pull the trajectory towards it, just like this. And this step will be repeated many times, and every time a new cell will be selected and it will pull the trajectory towards it. The trajectory will also be allowed to split from time to time, so that it can represent things that are more complex than a simple line. We also track the usefulness of each line, because if at some point a line becomes useless, this allows us to get rid of it. These simple steps allow to allow Tenga to identify trajectories quite accurately. See for yourself. The power of Tenga is that it can actually model any shape, which makes it quite powerful compared to other tools. Here, I'm showing that Tenga performed quite accurately in these different shapes that I showed you. Whereas other tools, such as slingshots, you can see that it is actually a very good tool to identify trajectories that are quite simple in this case. But the tool is not that accurate when it needs to identify complex trajectories. You can see that Slingshot found this blue line over here, which is very different from what it should have found as a structure in this data. On the other side, we have tools like PAGA that are known to be very efficient to identify complex trajectories, but these tools typically tend to identify two complex trajectories on simple data sets. So in this case, it should have found a Y, and you can see that what PAGA identified is actually much more complex. So this is the strength of Tenga, not being biased towards what it finds. In summary, we've seen that when we have this type of data, we can learn how to represent it in reduced dimensions. And then in these dimensions, we can identify a trajectory. I would now like to show you some applications of computational tools, and first we'll focus on the generation of memory cells. As I mentioned in my introduction, memory cells are cells that patrol in our body and confer us protection. If the same virus would show up again, these cells are able to get rid of it very efficiently. This is very useful because many vaccines rely on the generation of efficient memory cells. And so understanding how to generate better memory cells can help us to produce better vaccines. In this study, we worked with data that was generated by infecting cells at what we called day 0. And then we sampled T cells that were generated in response to this infection. And we took them either at day 4 or day 7 after infection. Then the copies of more than 2,000 genes were measured in these cells, and this resulted in these data sets. We fed this data set into Tenga, and this is the trajectory that we obtained. You can see a nice transition from T cells from day 4 to T cells from day 7, with a little split here at day 7. As I mentioned in the introduction, a trajectory is very interesting because it also allows to identify features that make sense in this trajectory. In this case, for instance, we were able to identify MKI67. This gene is represented in this plot by its amount. So if a cell had a lot of copies of this gene, then it was represented in yellow or red. Whereas if the cell had no copies of the gene, then it appeared in blue. So you can see that the great majority of cells that have MKI67 are here at the bottom of the trajectory. And we know that MKI67 is a gene that is being expressed in cells that are multiplying. So this informs us on the fact that this part of the trajectory nicely shows us how cells transit when they start to multiply. Another interesting gene was TCF7, which was clearly more expressed in the cells that were located at the top of this trajectory. And we know that TCF7, among other genes, is associated with memory. And so we knew that these this part of the trajectory, which was showing us how cells transition when they become memory cells. We were also able to extract gene regulatory networks at the single cell level and at the group of cells level, which allowed us to show, for instance, that these cells that were becoming memory cells had this gene regulatory network associated, which showed us which genes were actually actively being regulated in these cells. So we were interested in memory cells, and this was where they were located. So we looked at them in more details, and we saw that some of these memory cells were produced already early at day 4, but that they were relatively immature. And that we also had memory cells that were produced later at day 7, and that these cells had more of a mature profile. They were expressing more functional genes associated with the immune functions. So in summary, we saw that after a viral infection, the majority of cells would multiply and start producing these immune functions that allowed them to attack the infected cells and get rid of them. We also saw that a part of the cells actually stopped multiplying quite early and didn't really go into the fight, but they were rather here to stay for a later time point and confer us this long-lasting protection. Among these, we've seen that we had 2 different types of cells generated earlier or later after infection. And now this is interesting because if we found out that these cells have a different capacity of protection, then knowing which genes are being expressed in them, how we can push the cells to become either this type of memory or this type of memory, can allow us to produce better vaccines. Finally, I'd like to move to this third project that's in which we were studying the mechanisms of tolerance. So in this study, we were interested in patients with leukemia, which is a tumor that attacks the bone marrow. The bone marrow is situated inside of our bones. Today I've shown you several types of immune cells, but what I didn't tell you yet is that they are all being produced in the bone marrow. So patients who have leukemia are typically quite weak. They are not able to defend themselves against the disease. A last resort solution consists in giving them a chemotherapy treatment, which will have the effect of removing their immune cells and then transplanting them the bone marrow of a healthy donor. If everything goes well, the new bone marrow will start producing cells that will be able to recognize the tumor and get rid of it. Unfortunately, in many cases, the new bone marrow starts producing cells that actually recognize cells from the patient, which results in the damage of skin, liver, mucosal membranes, and typically the prognosis of these patients is very poor. So in this study, we had 2 types of patients: tolerant patients who were protected by the new bone marrow, and non-tolerant patients who were actually attacked by their new bone marrow. And this slide might remind you what I showed you at the very beginning, where we had these 2 images of 2 different types of patients, and we were trying to classify them. In this case, we didn't have images for these patients, but blood samples. And in these blood samples, we measured gene copies in the cells, but we also measured proteins and metabolites. We also had clinical information on the patients, so we knew for each patient his or her age, gender, the age of the healthy donor who gave them the bone marrow, and of course most of the time the compatible donor was a sibling, etc. So for each patient we had these clinical information, and then we had 3 different types of information. How many copies of these different genes they had, what type of proteins they had, and the amount of metabolites that they had. Of course, we also knew for each patient if they were tolerant or non-tolerant, and this is when we started looking for associations. For instance, in this table, you can see that this red gene is much more present in non-tolerant patients than in tolerant patients. So this gene copy might be playing a role in the disease, and so it was quite interesting for us. If we look at this yellow gene on the other side, you can see that it's present almost in the same amount in tolerant and non-tolerant patients. So these are typically genes that we didn't focus on. If we looked at the clinical features, there was one feature that really came out significantly, which was that younger patients had statistically more chances of being tolerant than older patients. So just as a quick reminder, in this study we had tolerant and non-tolerant patients. These tolerant patients who started producing cells which attacked the tumors, and non-tolerant patients who produced cells that actually attacked them. And so this is what we found as variables that were really more present in tolerant patients. So here are the metabolites, gene copies, and proteins that were more present in tolerant patients. And we saw that all of these molecules were typically associated with an anti-inflammatory state and an immunosuppressive response. So all of these molecules were really helping to keep the patient's immune system under control, like not too reactive. On the other hand, these are the molecules that were more present in non-tolerant patients. And on this side, we had a lot of pro-inflammatory molecules that were activating the immune system. So this showed us that the immune system of these patients was still heavily fighting. So in this study, we found out that tolerant patients were typically younger and they had more of these anti-inflammatory molecules, whereas non-tolerant patients were typically older and had more inflammatory molecules. And so we found a certain amount of molecules that could explain this. And the idea now is that we study these different molecules that we identified into more detail, because they might give us the possibility to, at some point, develop drugs that would prevent patients from being non-tolerant to this graft. So in this presentation, I've showed you Tenga, a tool that I developed to identify trajectories in single-cell datasets. We've seen how we could apply it to better understand how memory cells were being generated. And we've seen this last study in which we looked at the mechanisms of tolerance. So today we can measure many things in biological data. We can measure genes, proteins, metabolites, but also many other things. And this typically results in very large and complex data sets, which is why we now need computational tools to help us to analyze these complex and huge data sets and to extract knowledge out of it to produce, for instance, better vaccines or better drugs. I thank you for your attention, and I will now be happy to answer any questions you might have.